Hello everyone, my name is Pedro Diaz Payno, and I am a postdoc in Medical Delta. I work in the RegMed 4D project in Medical Delta, which stands for Regenerative Medicine 4D. It involves the generation of complex tissue engineering strategies, combining stem cells, 3D printing technology, and changes in time. The project of RegMed 4D involves the collaboration between 3 different universities from Leiden, Delft, and Rotterdam. In particular, I'm working in the laboratories of Professor Gerrie van Als in the Department of Orthopedics at Erasmus MC in Rotterdam, and in the laboratory of Professor Amir Thaatpour in the Biomechanical Engineering Department at TU Delft in Delft. And I focus on the tissue engineering of complex osteochondral interface for regenerative medicine and disease modeling applications. So why are we interested in the regeneration of the osteochondral interface? Articular cartilage is a soft tissue lining at the end of the bones in our synovial joints, and enables a smooth pain-free articulation. Cartilage defects show poor long-term regeneration, leading to pain, impaired mobility, and can progress to the subchondral bone, developing an osteochondral defect. If untreated, such defects can progress to osteoarthritic joint degeneration, a disease which affects millions of adults worldwide. Articular cartilage and osteochondral defect repair still remain a significant clinical challenge, and new tissue engineering strategies aim to tackle this condition. But what is tissue engineering? Tissue engineering can be defined in this triad in which we aim to combine cells that can be isolated from a patient, regulatory signals such as growth factors will orchestrate the tissue that the cells are depositing, and the biomaterial onto which these cells can be seeded to produce or engineer the tissues that can be transplanted or used for disease modeling applications. In tissue engineering, there are different approaches, but in particular in our osteochondral tissue engineering, we can follow 2. The first one is to look at the native tissue and see how it's constructed, how it's fabricated, how does it look. Articular cartilage and bone have very different properties. They vary in cell type, cell density, and it changes in stiffness. On the other hand, we can look at how the tissue develops. So bones are formed from a very soft cartilage template, which over time increases in stiffness. So with that in mind, I have 2 research questions. Is how can we fabricate constructs that recapitulate the complex gradients that appear between different phases in native tissues in the osteochondral interface. So for that, we can use bioprinting, which allows us to spatially control material and cell deposition. The generation of different zones is possible by using tailored bioink compositions, which can be based on alginate, hyaluronic acid, collagen, extracellular matrix, and other hydrogel-based materials. And we can create these structure layer constructs for the generation of different zones. Challenges associated are the assembly of layers without delamination and biocompatibility. All these can be summarized into choosing the right bioink. Question is how can we modulate the properties of such constructs in time? So first we need to understand what's 4D. 4D refers to structures that can shift between at least 2 stationary states. This can be useful to obtain advanced functionalities over time. The dynamic behavior needs to be predictable or controllable when triggered by an external stimuli. And so this is using the so-called smart materials, which are stimulus responsive. In order to modulate the properties of such construct, first they need to be 3D printable smart materials, and by that they can be stimuli responsive polymers that either have shape memory properties, self-morphing or self-reconfiguring, and are triggered by stimuli. And we can encounter different stimuli. And some of the examples can be physical, which is it can be triggered by light, chemical by pH changes in the media, or biological when their materials are functionalized with semantic specific sites. Another important factor is the rational design of geometry of the scaffold and the internal structure. Proper combination of the smart material, the stimulus, and the rational design of the construct's geometry guarantees the success of the 4D process and generation of materials with impressive properties. This project is a national and international collaborative research and involves many organisms and institutions, which all try to go forward in the regenerative medicine. Thank you very much for your attention, and hope this has helped you to understand a bit more about the RegMed 4D.